day to everyone, and thank you for registering to this live webinar. We're hosting this webinar with attendees from all around the globe. Welcome, everyone. My name is Ricardo Suarez Heredia from University College London, and I will be hosting this webinar on behalf of the Early Career Members Network of the Metabolomics Society. This is actually the 17th of our webinar series session, and it's brought to you by the Early Careers Member Network Committee. Currently, the webinars are free for all to attend and by the courtesy of the Metabolomics Society. And I would like to inform you that all the materials will be recorded and uploaded to the Metabolomics Society website, allowing you the access of the live content at your own convenience. Please connect with us with the Metabolomics Society and the EMN on Facebook, Twitter, and LinkedIn, as shown in the screen. I would also like to ask you for feedback about the Early Career Network. You can share your ideas and suggestions on these initiatives at the email address info.mn@metabolomicsociety.org. Before introducing our speaker, I would like to introduce you to the Metabolomics Forum once more time. It's a platform where people can talk about or discuss any subject related to metabolomics, Metabolomics Society, hardware, software, databases, troubleshooting. If you have any question related to metabolomics, you can post it on the forum and get answers from other users who are experts in the field as well. Please visit the forum, create your own profile, and use this platform to talk and discuss metabolomics. The link is displayed on the slide. The annual International Conference of the Metabolomics Society as well is coming up from June 24th to 28th of June in Seattle, USA. The network is going to be actively present, and something I would like to highlight, the abstracts call is still open, and the deadline for oral presentations is on Wednesday. For poster presentations is in April. Please check the details on the website. I would like to inform you as well, you can find the agenda for the overall conference, including 15 workshops that have been scheduled for during these activities. The network is going to be present, and we will be delivering some workshops for all our members. I would like to introduce the speaker. Today's speaker is Dr. Nathan Lewis, who is an assistant professor in systems biology at the Department of Pediatrics at the University of California, San Diego in the USA. His research is focused on the integration of omic data with in silico models of metabolism and tool development for constraint-based modeling. Particularly in his lab, he integrates these approaches to map out and engineer the cell pathways controlling mammalian cell growth, metabolism, protein synthesis, and protein glycosylation in an effort to develop improved drug production, host cells, and unravel complex mechanisms underlying human diseases. There will be time. I would like to mention that there will be time after the webinar for questions. So please send your questions by using the question option on your webinar application. You can type these questions throughout the presentation, and our speaker will try to aim to answer as many as possible in the remaining time. I think we're ready to start, and I will pass you on to our webinar speaker, Dr. Nathan Lewis.
Okay, so thank you. Thank you for attending. Today I'm going to talk a little bit more about my work. In particular, this is going to be a little bit different from your typical research talks in the sense that I'm going to talk about some of the different tools and the abilities that you can that there are for analyzing metabolism comprehensively in a cell. And so basically, I don't need to tell too many people about this, but the metabolism is actually quite fundamental. Now, these are some pictures painted by David Goodsell, in which he's showing right here the metabolism of, or rather the inside of a mitochondrion. So you can see the pyruvate dehydrogenase complex and many other different components in there, then this ends up being fundamental to all living processes that are seen. The activity of metabolism is essential for producing building blocks and also for fueling everything that's going on inside of a living organism. Now, when you're interested in metabolism for many reasons, it can be just due to one small metabolic defect that a child can have, growth delays. Also, if you look at cancer, cancer is studied for many of its hallmarks, ranging from its impacts on angiogenesis, to disabling apoptosis, to perturbed cell adhesion and metastasis. Now, one thing that people don't often think about is that metabolism is connected and plays an intimate role with all of these different hallmarks in cancer metabolism. Then you can go beyond that and you say, well, the gut microbiome, its metabolic capabilities significantly impacts and interacts with all of the different human tissues. And so metabolism is incredibly important for all these different interactions in the microbiome, the human body, our growth and health, in addition to all the many diseases that I've mentioned. So one question we have is, how can we comprehensively study cell type and tissue-specific metabolism, given that its importance and its role in so many diseases and normal physiology? Well, so tissues and cell types add in some complexity in that each tissue and cell type actually specializes their metabolism to be able to accomplish specific tasks that the organism needs. And so one could go through and actually look at the various tissues and for how they all interact. And so there's a computational model built a few years ago in which they did exactly that. They looked at how all these different tissues interacted with their metabolism to be able to look at drug metabolism, biomarker identification, and many other phenotypes. But when you start thinking about metabolism, it is quite vast and complex. Now, this is just a snapshot, a portion of the metabolic pathways represented in the KEGG database. And so when you start thinking about the great tools we have in studying metabolism in metabolomics and fluxomics, those tools are quite powerful, and we use them a lot in our lab. But sometimes you want to get an entire view of metabolism in a given cell type or tissue, and it's not immediately clear how to do that. Metabolomics is great. You can measure hundreds, maybe up to a couple thousand different metabolites in a cell, but we're still working on ways to make it more comprehensive given the different chemistries of the different metabolites. And then fluxomics itself also is typically focuses on a subset of pathways in any given analysis. So the question is, can we comprehensively study a cell's metabolism? Well, how do we do this for other processes? Well, pathway enrichment is a commonly used technique for being able to use RNA-seq or transcriptomics or proteomics to comprehensively assess the pathways, at least which overrepresented or significantly changed. But some limitations of pathway enrichment is that while it gives this excellent overview of differentially regulated metabolic systems, it doesn't assess the pathway function itself. Basically, what you're going to look at is you're going to see that an overrepresentation or an underrepresentation of various changes in a given pathway. And so what we were wondering is, can we get a better picture of a cell's metabolism? Well, I'm going to talk about two different ways that you can go about doing this, and some work that we've done on both of these fronts. So first, you could look at, instead of just looking at generic pathway groups, you can actually take a pathway with its inputs and its outputs and assess the expression of all the different steps to see if it's likely that metabolic function is going to be active in a given tissue. So not just pathway enrichment, but actually assessing whether or not the function of a pathway will be achievable in a tissue and be able to compare that to other tissues. And then second, You can also simulate the metabolic flux if we can build accurate models of the whole cell metabolism or the whole tissue metabolism. So I'm going to talk about about work we've done in both of these areas to kind of give you an idea of how we can start to get a more comprehensive view of metabolism that will supplement powerful metabolomics and fluxomics studies. So first, to be able to do this, you have to have a comprehensive view of what metabolism looks like for your given organism. And the way you can do this is you can go through and you identify all the genes that are known to be metabolic, and we can go through and tap out the decades of research on metabolism to be able to connect each of those reactions to metabolites that are being acted upon by the enzymes. And that gives you basically enzymes and the reactions they catalyze, and then allows you to be able to stitch together all of those reactions into pathways such as we see here for glycolysis or the TCA cycle and other pathways. Once you have that, then you can do all sorts of different things. These will capture all of the known metabolic pathways in an organism if done comprehensively, and this will include potentially thousands of genes and reactions and metabolites. And so you can then overlay your data on these and use various algorithms to assess the impacts upstream and downstream of any changes you make in your cell. And so there's several different databases that have done this or studies that have accomplished this in human metabolism. Recently, our group helped with other groups to put together Recon 2.2, but more recently, Recon 3D basically has brought together all of the previous work by leaders in the field, such as Bernard Paulson, Jens Nielsen, and Inez Thiele and others, to basically map out all the metabolic pathways and include protein structures. So right there, we have the resources to be able to look comprehensively at metabolism. But how do you use these? So what we're going to be doing, what I'm going to talk about first is how we can use this to figure out what is our tissue really doing. And to define that, we have made a definition, well, others have defined this before, of these tools called metabolic tasks. Basically, a metabolic task is given a specific metabolite uptake. How likely can a cell make other metabolites? So for example, you could have tryptophan could be taken up and through a couple metabolic reactions and start to make downstream metabolites that are important biomarkers for cancer or potentially for neurotransmitters. Or we can see right here, you might have more extensive metabolic pathways, such as the TCA cycle, where you have acetyl-CoA comes in to form all sorts of different metabolites through it. And so you can have these metabolic tasks. It can be a couple reactions, they can be larger pathways. But what you can do is then overlay data onto these to gain insights into whether or not these metabolic tasks could be achieved in your tissue of interest. So what we have prepared, or what we've developed, is a platform that should be easy for users to use, that allows us to capture a cell's metabolic functions. And what this does is it takes all the metabolic pathways in the organism and breaks them into these smaller components that allows us to allows people to overlay data onto them and define and quantify whether or not and to what magnitude different metabolic tasks can be achieved. And so this can include pathways that are associated with energy metabolism, nucleotide metabolism, amino acids, lipids, vitamins, cofactors, and so forth. And again, this allows one to quantify the propensity of a cell or tissue for a metabolic function. So we've gone through and curated hundreds of these metabolic tasks. What sort of processes are they covering? Well, a large number are covering amino acid metabolites, carbohydrate and energy metabolism, glycans, lipids, nucleotide, vitamin and cofactors, and so forth. And you can go in and zoom in more, like for example, in the vitamin and cofactor metabolism, you can have a number of other factors such as folate metabolism or heme metabolism and so forth. And so what you can do once you've defined all of these different metabolic tasks, you can test to see if your tissue of interest can do it. Now, if you look at the gene expression levels across many different tissues for a given cell, you can see that in some tissues you're going to have very high expression, some tissues you'll have very little or no expression of a gene. And so what you can do is you can say that these highly expressed housekeeping genes are considered active, the non-expressed genes are not. But what do you do about everything in the middle? And that's where a lot of the tissue-specific or cell type-specific functions reside. Is that what you can do is you can put bounds on everything that is very highly expressed or everything that's very low expressed and look in that middle to see which tissues is that gene more highly expressed. And that gives you an idea towards the tissues that are more likely to have these metabolic functions in particular. And so we're able to overlay the transcriptomic data onto all these metabolic functions in this manner to give a tissue-specific signature of the metabolic functions. And so right here we have a heat map showing the metabolic tasks for dozens of tissues in the Human Protein Atlas. As you can see, there are certain metabolic tests that are very active across many different tissues, but again, there's some tissue specificity. You have others that show lower expression, but still a preference for one tissue or another. And so one thing we were wondering was, do these metabolic tasks the organ system in which they're being used? So the idea here is that the organs themselves are going to have a specific metabolic function that they will achieve, and they've partitioned it across the different organs. And so sure enough, when we run the simulations to look at the metabolic tasks across all tissues, you can see that that the tissues themselves seem to cluster together based on their metabolic tasks, such as the lymphatic tissues, the female reproductive tissues, and the gastrointestinal tissues. We have the upper GI and the lower right down here. And so so one might consider, well, what are these tissue-specific metabolic functions? And we found that actually these clusters more significantly than you expect by random chance, suggesting that there is conservation of metabolic functions within organ systems to some degree. But what we're more interested in is the cell types themselves, and because the cell types themselves are going to define what the tissues are doing. And what you can do is you can go and look at the histology of each different tissue and define the types of cells that are in each of these tissues. So as you can see right here in the esophagus, you're going to have mucus cells or submucosa or mucosa and so forth. You can do this for all the different tissues and then cluster them and see which tissues are more similar to each other. So for example, you can see right here, the small intestine and the duodenum are pretty much the same, and the duodenum is part of the small intestine. But those are the next most similar tissue is going to be the colon and so forth. Now, this is what you get if you cluster tissues by histology. Well, what about if you cluster them by metabolic type? Again, you can see over here, you have the small intestine and the duodenum are right next to each other. And so, let's see. So you have the small intestine and duodenum are right. Closest is the colon and then the rectum. And so you can see right here, we are getting the tissue-specific, the histology clustering is represented in the metabolic functions. So this metabolic function analysis is providing us a detailed insight into the unique functions of different tissue types. And so the next question is, can these metabolic tasks capture tissue-specific metabolic functions? So just for example, you can look at serotonin biosynthesis. Now, serotonin is found predominantly in the GI tract, in the blood platelets, and the central nervous system. Well, what if you look at metabolic tasks? What about serotonin biosynthesis? Now, if you look across all the different tissues, you can see that that it is found predominantly in the GI tract. So you have in the stomach being produced, the rectum, and the duodenum in the colon. Now, something that's painfully obvious is we're not seeing blood platelets. Well, it just so happens that it's not produced in blood platelets. is produced by enterochromaffin cells that are predominantly in the GI tract. And so what's happening is the platelets themselves are soaking it up from the blood after it is produced by these other cell types. So the metabolic task analysis itself is actually able to accurately capture the cell types that are specifically producing serotonin. And so we wanted to go in and look a little bit more into cell-specific metabolic tasks, in particular because if you look in this previous analysis, we didn't see the brain also producing it, but we do know that serotonin is actually produced in the brain by very specific and rare neuron types. And so in this bulk transcriptomic analysis, it was unable to capture the signal from those single cells. And so we wanted to look at single cell RNA-seq to see if we'd be able to capture these cell-specific metabolic tasks. So we gathered data from a study in which they did single-cell RNA sequencing on 466 cells from the human hippocampus and cortex. And through this, they were able to cluster the cells based off of their expression profiles and identify the different clusters that were associated with endothelial cells, neurons, astrocytes, and so forth. And again, they had samples from many different cell types and 6 different ages. And so what we did, we were wondering is, are there cell-specific metabolic functions? It's been studied quite a bit in the brain, and clearly there are. I mean, you could have different neurotransmitters, you have different metabolic activities for different cell types. And so what we did is we took the RNA-seq data and we defined the thresholds and then partitioned the metabolic tasks to the different cell types to see which cell types preferentially did all the different metabolic functions. So what did we get? So just as an example, we're going to first compare neurons with their astrocytes. Now, neurons are typically not interacting as closely with the blood vessels. You have these astrocytes that mediate a lot of metabolic functions and also support the neurons to ensure that they have the right metabolic environment for effective functioning. And so we quantified the metabolic tasks for all the neuron cells and the different and the glial population. And sure enough, what you can see right here is the cells clearly cluster by cell type in the into neurons and astrocytes. So clearly demonstrating that yes, there are unique metabolic functions captured by the 2 different cell types. So what are these? Well, let's first look at the neurons. Now, the neurons are going to require the generation of a large amount of ATP to be able to fuel the neurotransmission and also to aid in the biosynthesis of neurotransmitters and the recycling thereof. So what did we get? So here is a list of different metabolic tasks that were neuron specific. And so what you can see right here, you can see that that there are a number of processes for synthesizing specific neurotransmitters. We're seeing, for example, serotonin, histamine, beta-alanine, dopamine, and nitric oxide, for example, where these are specifically enriched in the these metabolic tasks are specifically active in the neuron population. But we also found that to generate all this ATP, we have a dominance of oxidative phosphorylation and also the development of precursors needed for effective redox metabolism. But again, the neurons themselves are not alone in the brain. You have these astrocytes that are playing a supportive role. And what we see, and it just so happens that there's a number of different metabolic functions these are doing. For example, recycling neurotransmitters, storing glycogen for and utilizing glycogen to be able to fuel to provide metabolic fuel for the neurons as needed. And then also to drive something called the lactate shuttle, where it consumes glucose, produces lactate, and then provides that to the neurons as fuel. Now, some of these things are a little bit debated in the field, but we wanted to see, well, what about the metabolic tasks of the astrocytes? What are we seeing? And so what you can see there is that we are getting the lactate shuttle. The metabolic task analysis actually demonstrates that astrocytes are likely producing lactate for the neurons. We're also seeing the recycling of neurotransmitters and also the metabolism of glycogen for the neurons. So what we're seeing here is the metabolic task analysis is allowing us to now get down to cell-type specific, cell-specific metabolic functions. And so in summary, for this first part, we're demonstrated that we've developed this platform and established it and provided in a easy to use framework that people can just overlay their transcriptomic data and get a comprehensive view of the metabolic functions of a cell. Now, this approach does capture the unique metabolic features of different organ systems and tissues, but also it can be used to capture cell-specific metabolic functions. So this should be a nice tool to as a supplement to metabolomics and fluxomic studies, in addition to typical methods used for transcriptomic analysis, such as gene set enrichment analysis and so forth. But again, this approach, instead of just going for an enrichment of gene sets, now actually gives you a function of the cell. So that gives you a very easy to use approach. Well, how about something a little bit more complicated? Oftentimes, we don't only care about the metabolic functions, but we care about the metabolic fluxes and being able to get an idea of how they differ from one cell type to another cell type or one tissue to another. So the question is, can we accurately model metabolism from an expression profile? Again, going back to what I talked about earlier, you can build these genome scale metabolic networks models by stitching together all the metabolic enzymes in a cell. And then you can use algorithms to assess the impacts upstream and downstream of any lesion or any changes in expression. So for example, you can be able to predict growth or yields because all of the materials needed for cell growth are metabolic products. And then once you start bringing in the uptake and secretion of various metabolites, typically used obtained from metabolomics data, you're able to run simulations to see how quickly can your cells grow, or how active are different metabolic functions, or which pathways are being used, or the effect of different perturbations. So for example, if you knock out enolase, how is that going to, or you knock it down, how is that going to affect everything downstream? We have simulation, we have algorithms that allow us to be able to simulate that and gain insights into that. So, so how do you do this? How do you run these models? Well, there are a number of tools out there, just as a plug for a couple of them. There's OpenCobra is a community in which a few of the tools have been provided that for running the simulations, building and simulating with these models in MATLAB, Python, or other environments such as Julia. And so if you want to know exactly how to do this, there's actually a great protocol out there. The Cobra Toolbox, the MATLAB version, has a protocol that is currently accessible in arXiv. And with that, it can actually walk you through how to build tissue-specific models, for example, and run simulations therein. This is a great effort put forth started by Bernard Paulson, but taken on and being really led well through Ronan Fleming's lab out in Luxembourg. And so how do you build a tissue-specific metabolic model? Well, first, you have to have a genome scale metabolic model. Then you overlay your expression data. Now, not all genes are going to be highly expressed, and so you might have something where there's going to be a number of gaps in your network. So we have various algorithms that have been proposed by many others, where you can take phenotypic data or assumptions of an algorithm to be able to fill the gaps and to get a model that can carry flux. Now, the challenge is that depending on various decisions that you make in the process, you might get different models, slightly different models based on the expression data from your tissues, or in particular, the parameters you use to set up the model. So how do you do this? Now that in the development of a tissue or cell type specific model, you have several decisions to make. The first is you need to choose which algorithm you're actually going to use. Now, the challenge is that there's many different algorithms out there, more than a dozen now, and this is just screenshots of the papers that have presented some of a handful of ones that have been used by many people. So you have to choose which method to use, which one's best. Well, the way it usually goes is the one that's best is usually the one that's easiest for you to get access to. Rather that's the way a lot of people move forward, or you develop your own algorithm, and that's happened several times too. But we really need to understand what are the pros and cons of the different methods and which ones perform better for which different types of analyses. Many of them are great for different types of analyses. But then you also have to decide how do you define whether or not a gene is considered on or off? This is a histogram showing the transcript abundance of a cancer cell line. And you can see right here, there's definitely some highly expressed genes and there's some lowly expressed genes. And there's going to be genes that aren't really expressed at all, that aren't even included on this. So the question is, where do you draw a threshold of whether or not a gene is expressed or not? Is it up here? Is it a highly expressed genes? Or is it down here where all the expression, where you're basically seeing essentially no expression? Then you also have to decide how do you constrain your model? Your cell is going to be able to take up a certain number of metabolites and secrete other ones. And so can you use that information either experimentally, or can you just assume that the gene expression will decide that for you? So what you do is you take your model and you have to figure out what metabolites can go in and which ones can go out. This can either be defined by using anything that has a transporter, or you can use metabolomics data from spent media to predict or to be able to assess the direction of uptake or secretion and also the magnitude based off of metabolomic changes over time. So these are three very important decisions to make when you're developing these models. Now, the hard question first is, do these approaches actually accurately predict the metabolic fluxes of a cell? So there was a study a few years ago that called this into question. In this study, they took several different algorithms to predict the metabolic fluxes, to take transcriptomic data, to integrate it with these genome scale metabolic network models for E. coli and yeast, and to see if they accurately predicted the metabolic fluxes as measured using your standard fluxomics metabolic flux analysis approaches. And so one thing to look at here is you have these various different algorithms. Right here, the first one is PFBA, which is an algorithm that just doesn't take in the transcriptomic data. It's the only one here that doesn't. And it just assumes the cells are going to metabolize your substrates as efficiently as possible. And what we get right here is that the difference between the predicted metabolic fluxes when not using any transcriptomic data at all typically is actually better than the cases where you actually overlay the transcriptomic data. So this raises a question. Has E. coli and yeast just significantly enhanced their metabolism so that any algorithm assumptions actually decreases the accuracy of these models? Or maybe the fluxomic data is computed from an entire model and isn't taking into account gene expression data, and that's a limitation. Well, the question is still unclear, or the question is still unanswered in that regard. But one thing we do know in human metabolism, for example, there clearly is specialization of metabolism. Certain genes are activated or repressed. And so we're wondering if these algorithms improve the simulation predictions in human tissues. And so, just as a little bit of background, I'm going to talk a little bit about some of the different algorithms that are used to build these tissue-specific or cell line-specific models. So one of them is GIMI. Now, GIMI, this algorithm, what it does is you define what the cell is going to do. For example, is it going to grow? Well, then it has to produce all of the metabolites needed to build cell membranes, proteins, provide energy, and so forth. And so what this algorithm will do is it will take the transcriptomic data, overlay it onto the model, and minimize the amount of flux through reactions that show poor expression. So for example, this reaction right here might be poorly expressed, but it is needed to make acetyl-CoA. However, PFL is not needed to make acetyl-CoA because you have pyruvate dehydrogenase that is highly expressed. And so basically what you'll get is a model that is most consistent with the gene expression pattern. There's a other class of algorithms such as IMAT and INIT, and these ones minimize the addition of reactions that have low gene expression while maximizing the addition of reactions that show high gene expression. So it does not make an assumption about the requirement for growth. Another set of algorithms, such as FastCore, MBA, and Mincadre, in those you predefine a set of core reactions that have to be active. And then what it does in one way or another, it minimizes the number of lowly expressed or non-core actions to get a functional model. So what we did is we took Recon-1, so a genome-scale metabolic network reconstruction developed by the Bernard Paulson's group, and built 3 input models based off of metabolomics data. And that is basically in where each of these models, one of them, we left all of the transporters open, allowed it to take up or secrete any metabolites it needed. And then we used metabolomics data to constrain either directionality and magnitude of the uptake and secretion. Then what we did is we took, for 4 different cancer cell lines, we took RNA-seq data, overlaid it, and using different expression thresholds and also different algorithms for integrating those data to develop 288 different models with variations in parameter and setup. And we then use a few different approaches to benchmark these models to see which ones were behaving better than others, to get an idea of the most important decisions you're making when building these models. So the first question was, do the decisions on expression threshold, the algorithm, or model setup actually make a difference? So again, we took the different input models with the different constraints and overlaid the RNA-seq with the different thresholds using multiple different algorithms. And sure enough, what we see for these 4 different cancer cell lines, you could get models that only had a few hundred reactions or models that had nearly 2,000 reactions. So clearly these decisions do significantly impact the content of your cell types or your cell type specific model. So which decisions actually influence your network, the size of it the most? Well, it's probably not too much of a surprise, but the first principal component when you're looking at these models is actually the threshold you use, whether you use only the top 10% of the genes in red, or include all of the genes that are in the top 50% of expression. And what you can see in the third principal component, that's when the metabolomics data starts making a difference, whether you constrain it or not. So clearly the threshold is the most important. But what you'll see is if you look at the contribution of each of those decisions to the model content, while the threshold explains most of the variance, most of the variation in the first principal component, the method you selected also played a significant role. And so we want to understand these a little bit more. But now, so I'm going to step back though. We don't always care as much about which pathways are in a model. We care with these models, we care a lot more about what can they do, what can they simulate, and so forth. And so what can these genome scale models do well? Well, as I mentioned earlier on, all of the material you need for cells to grow, all of the metabolic functions and so forth of a cell are captured in metabolism. And so early on with these models, they showed that they were able to predict metabolic functions. What we found what Bernard Paulson found more than 20 years ago was that with these genome scale models, you could have the cells take up glucose as the cells are growing, they're consuming a large amount of glucose And in the process, they're secreting fermentation products like acetate. And they were able to simulate this with these genome scale models. So being able to recapitulate metabolic functions is an important function, function of these models. Another study that they did was they were able to predict for many different knockout strains in E. coli. They were able to predict the oxytrophies, or the metabolite requirements for those mutants. And they found that they were able to predict more than 90% of the growth phenotypes accurately for all these mutations. So being able to simulate gene essentiality is another important power of these methods. So looking at these cancer cell lines, we were wondering, do the models accurately predict metabolic vulnerabilities of them? So what we did is we obtained data using CRISPR-Cas9 knockout screens for 4 different cell lines. Now, in each of these, what you do is you print the CRISPR-Cas9 guide RNAs on a microarray, and then you load them into a lentivirus and produce it, and then infect a population of cells so that each cell has gets a different lentivirus with a different guide RNA. And so each cell has a different gene knocked out. And then you can run selections on these and be able to predict and be able to identify which genes were essential, which ones were not. And then you can go out and you can sequence the guide RNAs to quantify the essentiality of each gene. So we did this, and we took the model predicted essential genes, shown here in blue, and non-essential genes, and compared it to the model predicted essential genes, where we looked at the essentiality from the knockout screen itself, the experimental knockout screen, and to compare these to see, did the model accurately predict different phenotypes for the essential versus the non-essential genes? So just to give you a first answer right here, all of these algorithms, all of the models, almost all of them, didn't really matter what your uptake rates were or how you constrain the models, which thresholds you use or algorithm you use. Almost all of them outperformed the genome scale models, demonstrating that yes, these algorithms provided more accurate models that were able to more accurately predict the essential genes in these cancer cell lines. So it's up here is more accurate the higher the number is. But for these next plots, the lower the number is, the more accurate it is. So what we found was that the threshold did have a significant impact on the accuracy, where more stringent thresholds gave you better predictions from essential genes, and then constraining your model modestly improved the ability for these models to accurately predict gene essentiality. But what we found to be most striking was that the algorithm you used actually had the biggest impact on the model accuracy. So there are certain algorithms that behave better for predicting essential genes than others. But of course, these were all implemented using the approaches using approaches that would allow us to compare them most consistently across the rest of them. But there were certain classes that behaved better than others. But again, fortunately, all of the algorithms again behaved better than using the genome scale model alone. So this is kind of in contrast to that previous study in E. coli and yeast, in that we were able to demonstrate that these algorithms are being able to better predict the metabolic activities of various of these different cell lines. And so again, we found that the method chosen ends up having the greatest impact on accuracy, but you can find through the search basically combinations of thresholds and algorithms that perform best at being able to predict gene essentiality. So as the last thing for these models, one thing that we oftentimes want to do is we want to predict the metabolic function. Now, in the first half of my talk, I mentioned how we were able to quantify the metabolic functions of a cell de novo. Now, this is quite important because each tissue or each cell type has a very complex metabolic network. And so you don't always know. In fact, you usually don't know all of the metabolic functions in a cell. Here we're able to define 56 metabolic functions that a cancer cell needed to grow. And we were able to build another several hundred models and demonstrate that many of these metabolic functions, most of the models were able to recapitulate. There was a handful, though, that quite a few models actually missed. The big question is, why were they missing these? One, for example, is phosphatidylal and inositol biosynthesis. Now, 39% of the models that we built could produce this metabolite that's absolutely essential for cell growth. And so we're wondering, why did some of them not do it? Why were some of them not able to produce it? Well, for 42% of them, the upstream precursors to that metabolite were not being produced. And so for that reason, you could not produce phosphatidylal inositol. And 19% of them, the reaction that actually synthesizes it, were missing. So what was going on here? Well, here is phosphatidylal inositol in red. I'm sorry, in the lower left-hand corner where it says pale. That metabolite takes part in a cycle in which it can be phosphorylated and so forth. And so what happened is that in several of these models, the one reaction that was part of that cycle was included because it was highly expressed. And so every other reaction in that cycle was also included. Now, the thing is, in this cycle, the same amount of phosphatidylal inositol that is produced is also consumed. So you don't have any net release of phosphatidylal inositol for biomass production. And that is because the enzyme, the CDIPTR, that produces phosphatidylal inositol, that would allow it to accumulate, is actually lowly expressed. And so it comes down to the threshold. So that was an issue with that case. Now there's another metabolic function, that is the metabolism of tryptophan to produce a number of important metabolites. This was included in most models. Now, why not? Well, we found that curinase was highly expressed, but everything else in the pathway was either not expressed, or it was missing a gene, or rather, it wasn't expressed, but it was either missing a gene or it showed low expression. So the algorithms basically assumed that it was not expressed and that pathway was not active. So one thing, this would not have happened if we were able to pre-specify and enforce the metabolic pathway. But a number of these algorithms also failed to add it because certain reactions here are put in just for model usage. So they'll have, and so basically the algorithms were removing some of these reactions because we didn't have genes associated with them. In other cases, the gene associated with a reaction was either not known or not included in the model. So it was also the algorithms would also penalize the models for actually including it in that regard. And so in that case, what we see, even high expressed genes, reactions catalyzed by high expressed genes, could be removed if the algorithms did not have enough support of all the upstream and downstream reactions too. So is all lost? No. Basically, what you can see now, we have an approach that is being currently under development, in which we're able to incorporate the different tools that we talked about in my talk to be able to allow us to build more accurate genome scale metabolic network models, or rather metabolic network models of specific cell types and tissues. That is by integrating the information on a cell's metabolic functions with the model generation. And so this work will be available soon for people to use. So in summary, pathway inclusion in these tissue-specific models is impacted significantly by threshold choice. Of these models is most impacted by the algorithm you use. And we found that while most metabolic functions can be identified based solely on RNA-seq, work needs to be done to carefully curate these models to ensure that they have all the metabolic functions that are known in those cells. And we're developing a framework to be able to do that. So this work was done in large part by Anne Rochelle, a postdoc in my lab, and Chintan Joshi, another postdoc, and then Short Optim, a master's student that worked with me in the process. And this is funded by gracious funding from NIH, the Keck Foundation, and the Noah Ross Foundation. So any questions?
Thank you very much, Mr. Nathan, for all the insights during the presentation. Indeed, we have a couple of questions that we will be addressing. Let me start with the first one regarding the first section of the talk. In real-world metabolomics approach, we do have missing metabolites or platform specificity of metabolites leading to imputation, etc. How these are going to offset the FBA or genome scale modeling?
Could you run that by me one more time?
Sorry. In real-world metabolomics approach, we have missing metabolites or platform specificity of metabolites leading to imputation, etc. How these are going to offset the FBA or genome scale modeling?
So that's a really good question. Now, because of the various the diversity of chemistry and metabolomics, sometimes you are missing metabolites that you have to impute. What these can do is, if you can capture the major metabolites, for example, if you when you incorporate flux metabolomics data into flux balance analysis simulations, typically we look at the spent media, and we find that we can capture the vast majority of the fluxes based off of just constraining these models. And so the idea that you're sometimes missing metabolites, we're finding that a lot of our simulations are quite robust against that. And so, but there's always I mean, missing potentially be a challenge. But the flux balance analysis allows you to kind of assume that even if you didn't detect a metabolite, you can still potentially have flux through those pathways.
Okay. The next question is, in a linear or cyclic pathway, when biomolecules such as protein and metabolites are up or down regulated without following a trend or pattern, how the interpretation gets challenging? In other words, does FBA and model building takes into account post-transcriptional and post-translational or other regulatory processes?
Yeah, so a number of those are very important dynamic attributes of metabolic flux. And flux balance analysis on its own doesn't really capture the dynamic aspect, although there are adaptations of it that do. There are techniques out there that allow you to account for some of these post-translational regulation, whether it be through metabolite feedback or post-translational modifications and so forth. My group has a paper out in bioRxiv right now where we incorporated phosphorylation and acetylation of enzymes into these metabolic analyses. And there are approaches to interpret that in that regard. But yeah, it's still very much an open area of research on how to account for this.
Okay. The next question is, can the intracellular data, I'm assuming metabolites, be integrated to the FBA approach rather than, for example, what you mentioned on spent media constraints, or the constraints of the model?
For intracellular metabolites?
Yeah.
Yes, there's a few different ways that have done that. Jens Nielsen's approach in it that I talked very briefly about was actually a way to bring in metabolites into model construction. And then others have taken various approaches to account for changes in metabolomics data using, for example, unsteady flux balance analysis from Bernard Paulson's lab, and others, many other approaches over the past 10 years. There are several, and I'd be happy to point people that are interested to some really nice studies that have done that. But in that case, it kind of gives you an idea, gives you insights into how the different pathways interact with each other as conditions change and you see changes in concentration and intracellularity.
Okay. The next question is, why not Cobra as MATLAB independent and open source for the research community? Is there any limitation on that?
Why not making it MATLAB independent? A lot of it comes down to the early days of flux balance modeling and so forth, and a number of it came out. A lot of it came out of engineering schools that had access to MATLAB, and so it was developing in that framework because it was just so easy to use, and there was so much to do. Or so many different functions and supporting functions and so forth. And so that's why it's developed predominantly in MATLAB. But for that exact reason that the question came from, there's been a movement to develop it in other frameworks such as Python. And so there's the Cobra Pi framework that came out of Bernard Paulson's lab. There's also a framework that was developed using R, and a few other languages too. So there are modeling frameworks outside of MATLAB that are open source and free to use, where you don't have to have any licenses or anything like that. But the most heavily used package right now is the MATLAB one, mostly in academic settings where the universities have the licenses. So.
Okay. The next question would be regarding the nutrient essentiality for cells. Can a genome scale model predict the nutrient requirements for obtaining specific metabolic functions or phenotype?
There are some studies that they've looked into that. In some cases that they've actually looked at the nutrient essentiality of a certain. Well, this has been done many times. Where you could use the essentiality or inessentiality of a nutrient to predict which genes are missing from your model. That's been done before. People have looked at oxytrophies. In my lab, we developed a model of the Chinese hamster ovary cells, and we looked at oxytrophies, basically the essential amino acids that those cells have. There was an excellent study from Eitan Ripin's group a couple years ago, where they looked at the metabolite requirement of a large number of different bacteria. So yes, you can, and it's not always 100% accurate. In fact, usually it's not 100% accurate, but it gives you a really good idea of what a cell type needs and can be used to guide further refinements of the model when it fails to predict essential nutrients.
Okay. Just a final question. Along the lines of your presentation, it's noted that blood as a carrier of metabolites is not a good choice for finding cell type and tissue-specific diseases biomarkers.
Oh, sorry, will you repeat that?
It's noted that blood as a carrier of metabolites is not a good choice for finding cell type and tissue-specific disease biomarkers.
Well, so blood is a great source for identifying biomarkers because you can see changes in the metabolites that are shed or consumed from globally. So that's in that case, metabolomics is great. Building a model or finding metabolic functions might be a little bit trickier because if you're going to identify the metabolic functions the way we talked about it, you'd have to do transcriptomics on the blood, which means that you're really just looking at RNA from immune cells that are in the blood. So you might not be able to get a great view of all the metabolic functions in all the tissues in that regard. Yeah, so in that case, the approach might not be good for diagnostics, at least as good as your routine metabolomic studies. But it's great for giving you insights into the metabolic functions issues if you have samples of omics samples from those various tissues. More of a discovery, biological discovery tool.
Oh, okay. Apparently, there's no more questions. So with this, well, if you don't have any other comments, I would like to finish the webinar edition.
No. I want to thank everyone for joining, and I hope they found this to be informative. Typically, I give my talks about our new and exciting scientific discoveries, but I felt that this would be a good venue to kind of talk to you about the how-to and to utilize some of these tools to gain insights into a cell or tissue type metabolism. I really appreciate everyone attending. Thank you.
Thank you very much, Dr. Nathan. Well, with this, once again, I would like to thank you for all who joined and for yourself. We have spotted, as we discussed before, the GoToWebinar platform apparently doesn't get uploaded according to the different time changes. So we have spotted that for this webinar. So this recorded webinar for the attendants that have just joined will be uploaded as soon as possible into the platform, and you will have opportunity to visit the content once again. If you have any questions, please email Dr. Nathan. And I'm sure he will be happy to support you. Any questions you might have. I would just to remind you that the next edition is going to be in April. Please just look for the advert from the Metabolism Society. And if you have any feedback regarding the EMN network or any other comment on the initiatives, please let us know any feedback or suggestion. With this, I would like to thank you everyone for the ones who attended and our speaker once again. I hope you have a good day regardless of your location.